is toxin, so it's the toxin-bearing strychnose. Curare basically inhibits nicotinic receptors. It inhibits the nicotinic receptors. So if the curare is blocking the receptors that are normally being used by the nervous system, and in this case, the nervous system is using acetylcholine to communicate with the skeletal muscle, the curare gets into the acetylcholine receptors and prevents acetylcholine from binding to the receptor. Right? That's what I mean by inhibits these receptors. It blocks acetylcholine binding. What that does is effectively shut down the skeletal muscles, which means it's a paralytic. Right? It would shut down your ability to do much of anything. Too much of this, and it shuts down the diaphragm, and you can't breathe anymore. As an aside here, kind of a sad aside story, initially people thought curare was a really good, I can't think of the term now here, anesthetic. They thought curare was a good anesthetic, and they could give people curare and then they could operate on them, and they didn't figure out until much later that the curare wasn't an anesthetic. It just prevented people from communicating the fact that they were in a great deal of pain. That makes sense? All right. So that's curare. And then I have here Atropa belladonna, that's atropine, and this Mandragora officinarum makes atropine and scopolamine. Mandragora is mandrake. If you, Harry Potter, the mandrake root, they pull them out of the ground and they scream. That's actually based on the Veda, which is a very old Hindu text, which is a medical text which refers to the ability of mandrake root to do all sorts of things. And there was a run on mandrake root, I can only assume because this lore came up around it, that if you went into somebody's field and stole their mandrakes, if you pulled their mandrakes out of the ground, they shriek and they would kill you. So in this textbook, there's actually drawings of a dog with a chain, and the dog is used to pull the mandrake out of the ground because dogs, I guess, were immune to mandrake shrieking. In any event, it's a really kind of an interesting, mandrake's an interesting story anyway. It's in the Bible. People felt that if you slept with mandrake root under your pillow, it increased fertility. Eventually, the Catholic Church banned mandrake. That's one of the things that Joan of Arc was accused of, was having mandrake in her possession. In any event, the toxins that are found in mandrake include atropine and scopolamine. Atropine derives its name from the god Atropos, who's one of the three fates. You can see her here with her little pair of scissors. One of them produces the cloth, the thread, I should say, produces the thread, that's Clothos. One of them measures it, that's Lachesis. And Atropos is the god that cuts your thread. That determines how long your life is and ends it at that cut. So atropine got its name because of its ability to kill people. But in any event, atropine inhibits the muscarinic receptors. And if I inhibit the muscarinic receptors, I'm inhibiting the parasympathetic nervous system. And if the sympathetic nervous system is what is trying to return you back to baseline, right? Decrease your heart rate, etc. If I block that nervous system, then the sympathetic is the only signal being received by these cells. So atropine should cause things like dilation of the pupil and increase in the pulse because it's knocking out the parasympathetic nervous system. Okay? All right, so I could target the release of the transmitter, or I could target the receptors for the neurotransmitter. And then another common trick to do with toxins is to target acetylcholine esterase. And if I can target that enzyme, I would destroy the ability of this enzyme to take apart acetylcholine, in which case the muscle would continue to receive the signal and receive it until the muscle became incredibly fatigued. Okay, so a few of these. This one here, Physostigma, produces this molecule called physostigmine. Physostigmine is not a nice one. It's an acetylcholinesterase inhibitor. Because it inhibits acetylcholine esterase. It's a molecule that's produced by the calabar bean. That's this one here, lots of that toxin in there. And then similar toxins are also in organophosphates. The organophosphates are insecticides. Sometimes people overdose on these insecticides because it gets in their system through their skin or they breathe it, and then you get similar responses. And people have also figured out how to make a nerve agent out of it called sarin. Okay. All right. So that's almost all I want to talk about with muscles. I have one more thing I want to do with you. Just checking the time on my camera. And that's to talk a little bit more about kind of muscle types. Certainly not all there is to it, but just the basics so that as you move up in your courses, you can talk a little bit more about these with a baseline. Okay? All right, so different muscle fibers contain different myosin. So basically the type of muscle can interact with actin quicker or slower. So imagine the speed with which those myosin heads interact with the actin. And that's determined by the rate at which the ATP is hydrolyzed. If the myosin can hydrolyze that ATP very quickly, it can interact with more actin binding sites over the same amount of time. If it's a slower ATP hydrolyzer, then you have fewer interactions between the myosin head and the actin, right? So that's the speed with which the interactions occur is based on the speed with which hydrolysis occurs. Okay? All right, so low ATP hydrolysis rates gives us what we call slow fibers. And then high ATP hydrolysis would give us what we call fast fibers. And the ability of these fibers to quickly hydrolyze ATP, quickness of the contraction versus the slow fibers, very much more slow. Right? That's one way to classify muscle fibers, based on the ability of the myosin to hydrolyze ATP. The second type of classification refers to its ability to manufacture ATP. This is how quickly you use it. This is how quickly you manufacture it. So there are fibers that have lots of mitochondria. And because they have lots of mitochondria, they can do lots of cellular respiration, and they can do lots of oxidative phosphorylation. So these are called oxidative fibers. So some of these muscle cells are loaded with mitochondria. They can use all those mitochondria to make a lot more ATP. The ATP is then used for muscle contraction. Okay? Okay? All right. Usually these types of muscle cells have a good supply of blood from the circulatory system because having the mitochondria doesn't do any good unless you've got a big source of oxygen. So the oxygen here has to be coming from the circulatory system to provide the ability of these oxidative fibers to work properly. So there's usually a pretty good supply of blood vessels nearby, and you also find lots of a molecule called myoglobin. Okay? This is basically an intracellular reserve of oxygen. So, the way this works is when the circulatory system blood comes next to the muscles, right, oxygen diffuses into the muscle tissue. The myoglobin picks up some of this oxygen. It's another respiratory pigment, but it picks up this oxygen and holds it. And as muscle contraction continues, the oxygen can be used up faster in the mitochondria than it can diffuse in from the circulatory system. At that point, the myoglobin begins to cough up its oxygen to provide the ability to make more ATP, despite the fact that oxygen is diffusing in slower from the circulatory system, because the cell has loaded up on oxygen bound up in the myoglobin. Tissue that has lots of myoglobin in it tends to be red. So these are called red muscle fibers. Okay. Okay. Other fiber, not much mitochondria, but lots of glycogen. Okay. These are referred to, well, plus the enzymes that can work with glycogen. These are called Glycolytic fibers. And these glycolytic fibers, they run out of oxygen pretty quickly. There's not a big supply of oxygen in the circulatory system nearby because they're not that heavily vascularized. And they don't have a lot of myoglobin to hold oxygen in reserve. So what they tend to do a lot of is just glycolysis plus that little reaction to get your NAD plus back, which we refer to as anaerobic respiration, and specifically in muscles we would call this lactic acid fermentation. So you have a big supply of glucose, which means even though you're not getting 36 to 38 ATPs, there's lots of glucose, so you can make up for that fact by just doing lots and lots and lots of lactic acid fermentation, which is lots and lots of glycolysis. Okay? All right, now, if we take these two concepts together, the first way of thinking about it is ATP hydrolysis, and the second way is, am I biased toward cell respiration or toward lactic acid fermentation? And based on that, we can come up with three types of muscle fibers. The first one is called slow oxidative. Oxidative. And slow oxidative fibers are long, steady, in terms of workload. Think flight muscles in a bird, or long distance swimming, or in us, the muscles associated with our posture. So if I've been standing here, right, for the last hour or so, and standing here, right, I have to maintain a certain posture. Even though my posture is lousy, I still maintain a lousy posture. Those muscles have to stay contracted. They're slow. They don't need to work very quickly, right? They work over a very much longer period of time than the faster muscles do, but they're capable of sustained muscle contraction over a longer period of time. They can make lots of ATP, which means they can do long sustained, but I'm not asking them to work very quickly. So if I'm a bird flying south for the winter, those are slow oxidative muscles. Your posture muscles or a whale swimming through water, those would be slow oxidative muscles. Then we would have fast oxidative. Fast oxidative are important for very rapid action. I'm getting lots of ATP out of these things because I have lots of oxygen in mitochondria. I get lots of ATP, but I'm going to ask these muscles to work very, very fast and burn through that ATP. These are movements like a rattlesnake rattle, right? That has to be done using these fast oxidative. In birds, when they trill, I can't trill, but when they trill, they're using fast oxidative. It's like that, that's trilling. Okay, and then finally, fast glycolytic. And the fast glycolytic fibers are ones involved in short sprints. Think like a cheetah. Right? These muscles can do this high ATP hydrolysis, but we're basically asking those muscles to do glycolysis with a lot of glycogen stored up. So fast movement, but very short, not sustained over a very long distance. Okay? Okay. All right. Next thing about muscles here. You should be getting the idea that oxygen is a problem. And if we go back and look at how these muscle cells are organized, it's probably the best view here, how these muscle cells are organized is we're packing in all of this protein in the form of myosin and actin, and we're basically glumping up the cell